We will wait for a couple of minutes.
Just can I start?
No, we'll wait. We'll wait for a couple of minutes.
Okay, sir. No issue.
Okay, so let me shut down everything in the phone. Now, can you hear me?
Yes, sir.
Perfect.
You are audible. Just a
Yeah.
minute. We will
Perfect.
wait for 5. Then we
Yeah,
will
yeah, yeah.
start.
Hello, madam. Swardha madam, we can start now.
Okay, sir. Okay. Hello all. I, Swardha Kame, welcome you all for afternoon sessions. Hope you all had your lunch. Now we will have move towards the afternoon first session, that is on advancement in in situ hybridization by Dr. Enrico Otto sir. So I request Dr. Leena Pathak ma'am to introduce our speaker. Over to you, ma'am.
Thanks, ma'am. Thanks. A very good noon to one and all. First of all, I thank Dr. Praveen Badhe, sir, Swalife founder, for giving us an opportunity as an academic partner in this second international conference. I, Dr. Leena Pathak, Principal, Jain College of Pharmacy, on behalf of organizer, heartiest welcoming Dr. Enrico D. Otto, sir. Welcome, sir. Who is the Dr. Enrico Di Otto, who is the founder of OACPI Limited, Italy. Dr. Enrico, sir, is having more than 18 years of experience in cancer diagnosis that brings unique insights from his everyday experiences with less diagnostic tools. His expertise in oncology and clinical pathology, honored from Bologna University, and entrepreneur experience across USA, China, Asia to empower his innovation. Dr. Enrico's OACP technology, a novel chemical technology that overcomes current DNA test limitations, enhancing efficiency without altering existing lab procedures and equipments. Today, Dr. Enrico D. Otto delivers a talk on topic advancements in situ hybridization. Heartiest welcome, sir.
Thank you. Thank you very much. Good afternoon, everyone. So I will try to share my screen. Okay. I don't know if you can see it. Just let me know if you can see the presentation so I can go ahead. Can you see it?
Not yet, not yet.
No,
Not
sir.
yet. So let me try in another way. Okay. Okay. Maybe now, yes.
Yes.
Okay, great. Thank you very much. So again, hello again. Thank you for having me here this afternoon to just illustrate you something about the in situ hybridization, which is something that I did and I'm doing since a lot of time ago. Let's say this is me. I'm already been have been introduced, so I don't go through that. Yeah, basically for just for everyone to remind you something, in situ hybridization is a very powerful molecular biology technology that basically match specificity and sensitivity of the molecular biology with the possibility to directly observe chromogenic aberrations and genetic amplification, deletion, rearrangements, uh, basically something that it is typical of classic cytogenetic techniques. On the picture, you can see just a very schematic view of how in situ hybridization works. So basically on a tissue sample, you can directly hybridate the molecular strength, which is labeled with fluorescent signals, and then visualize cell by cell where the and if the alteration is. Currently, let's say a lot of application for in situ hybridization are in surgical pathology. So for cancer diagnosis, prognostic informations, and information about recurrences for cancer patients, and in situ hybridization following the basic, scanning and symptomatic examination from clinicians. So basically, patients after symptoms commonly is moved to an X-ray or TAC or echography examination. Then clinician take a biopsy or suddenly a surgical specimen from the tumor, then is sent to the pathology lab, and they are performing several tests, including in situ hybridization. Currently, the situation is that the standard procedures are not aligned with the expected efficiency to allow patients to heal and to better respond to treatments, in particular because the delay there is intrinsic in the method. I will show you better later. In situ hybridization, as mentioned, there are two types. One that I prefer more, which is the fluorescent in situ hybridization. So you can see in blue the nuclei, red and green are the probes. So red is a pathogenetic gene, and the green is the centromeric probe, so a control. Or colorimetric. So you don't need the fluorescent microscope, you just need a standard microscope, and the pattern is almost the same. So in blue, you have nuclei, and the red and black signals. But despite those two different ways to evaluate reactions, let's say, so fluorescent and colorimetric, basically there are no so massive differences. So mainly is that the fluorescent in situ hybridization is like in the picture labeled as A, so direct labeled. So once the patient DNA match with the probe, you directly see the signal. But this minor, not so used in the clinical practice, there is an indirect fluorescence. So there is the probe, which is labeled with a marker that is detected by an antibody that carry the fluorescent dye. The colorimetric, so the CISH, is basically like the second one, like the B, but the antibody is linked with a peroxidase that need a substrate to generate a signal. So in that case, the higher sensibility you have in the A, because it is direct labeled, and the lower sensibility, but it is not so I call it lower sensibility, but not so low, obviously, in the indirect CISH, because you have much more steps and stuff that are attached to the probe. Currently, and now we are starting seeing innovation in the process. There are two ways to perform in situ hybridization. So the manual way, so when a technician do all the steps by pipetting and so on, and automatic or semi-automatic stainers. They are starting, let's say automation is something that starts something like 15 years ago, not more than 20. And so considering that the manual fish is something that started with the cloning and with the radiation detection, so it's far older than the automatic. As mentioned, this is the general process, which is as the same as for manual and the automatic. So the process remains the same. So the resected tumor is processed, put on the slide. Then there is the DNA matching, and then after 2 working days, there is the report. Just to give you a quick overview, so we can see where innovation can be performed. Manually, you have to dewax, and the waxing because commonly we are working with the paraffin samples. So, and the waxing is commonly performed with harmful reagents like xylene. So an innovation in the waxing is given by the introduction of alternative to xylene, which are not harmful or a treat for technician. Then you have heat treatment, enzymatic hybridization, post-hybridization. As I say, manual method currently accounts for 80% of the testing way, all the in situ hybridization. It is flexible and allow a customization of case by case. So bigger sample can be left more time with the enzyme, or smaller with less time. However, this flexibility, as you know, generate a huge variability, not only internal within the lab, the same lab, but between different laboratories in different regions or in different parts of the same city. So the way, for example, I used to do in situ hybridization manually in the hospital where I was working is a totally different protocol that the neighbor hospital that is 5 kilometers from mine, they are doing. So, and when you are a patient moving from an hospital to another, to compare results sometimes is tricky in that way. You can see a practical imagine. This is the same case processed with 3 different protocols, and one is not failed. It was too digested, so no result can be performed on the far left, as you can see. On the center, it is barely readable, so can be evaluable. On the right side, it is not evaluable because it was not digested enough. So, but the sample is totally the same. So this is how to give you an idea how the variability stand. Automatically is, so the innovation brings by adding automatic or semi-automatic methods is not only in the standardization of the entire process, but also because everything is inside the machine, so it is safer for users that are not exposed to chemicals, compounds. And on the other hand, despite the standardization and the automatic process allow me to clone protocols from an hospital to another. So we are doing activities with a big pharma company as a OSP with my company. So we developed the protocol for automatic in situ hybridization in France. Then we cloned the protocol in Italy, and it worked very well. And then we cloned the same protocol in Spain, obtaining same results. So, and manually, it is not so simple to clone a protocol in different in different laboratories, but automatically you just send the file, they install in the software, and everything is done with minor adjustments, obviously. But it is a huge piece of innovation in a technique that it is still 80% manually. However, because in situ hybridization is a method that is impacted by pre-analytical situations, so how samples are managed before reaching laboratories, it may increase the number of repetition compared to manual protocol, because a standard protocol, or in molecular biology, there is no one-size-fits-all protocol, so some adjustment is needed. Basically, as I say, which is the state of the art. So in situ hybridization is something that the first innovation was in the probably 80s, where a company called Encore from the US bring the relevation, the way to reveal signals from radioisotopes or radiation to fluorescent signals. And it was a very huge innovation because it's reduced the exposure of biologists and technicians to radiations. Then another company started, and we are still in the early 90s, bringing color instead of fluorescent signals, but with protocols that at least 3 working days at least, and they were a nightmare. I did once, but a real nightmare. So the innovation probably brings the possibility to check results with standard microscope, but on the other hand, negatively impact the laboratory usability. So I don't think it is a real innovation if it make a technician life worse than before. A massive innovation started, as mentioned, in the almost 20 years ago with the automatic process. And the first example of real innovation is a colorimetric technique called silver in situ hybridization, which is a colorimetric in situ hybridization where the color is given by the silver that precipitate where the probe is matched. It was introduced historically by Roche, the pharma company from the Switzerland, and it was fully automatic process. I worked on the first version of this silver in situ hybridization that at the beginning was a single color test. Then they developed the dual color test, which is the current used in the laboratories, and it is working with certain limitation, but it works well for case by case, let's say. After that, they give the first step to start automating processes. Despite the market penetration of automation is 20%, so consider the in situ hybridization market is 2 billion, so that means a lot of money are going into the automation. There is the continuous need for implementation, because developing an innovation in a lab environment is something way different to bring that innovation to a real working environment, because the variabilities, the number of variabilities in the real environment are so high that you need to continuously implement day by day once on the ground. But on the other hand, automatic innovation needs or very well-founded startup companies or universities, or to partner with giants like Roche, like Abbott, like Molbio in India, for example, that have and are bringing to the market those automatic machineries. So what I did, and what a lot of people are doing right now, is to innovate and improve the manual procedures. So why? Because it is easier to innovate in manual procedures. It costs less for startup and academia, and also is a bigger market share than automatic procedures. So where the needs are, first of all, as I mentioned, standardization and time cost reduction. So regarding the standardization, it is possible by manufacturers that introduced pre-assembly and standardized reagents that avoid the too many steps or the use of in-house made reagents. So I'm working with a Chinese company that have basically developed 2 reagents, and with those 2 reagents you avoid to do 4 steps, 5 steps, and to be exposed to xylene, to toxic compound, to use flow desk, and so on. So just a water bath, a heater, and just put those reagents there. Additionally, so it is a pretty simple. So the standardization is something more in that case for chemical engineering people. So what we did and what we are trying to do is just to solve the time cost issue, because at least those tests, because of they require manually the use of person time, but also the control of the quantities of reagents manually is not as accurate as a machine, so or optimized as for the machineries. We need to improve efficiency of those tests, so it's still an issue, and we did it. Basically, my example, so it's just one, but because I did, I show you. Because the crucial step of in situ hybridization, as mentioned, is the matching of biomarker with patient DNA, and which is the step that is an overnight step, so it's a pretty long step, and the main costs in those tests are given by the cost for biomarkers. We basically developed the chemistry, as mentioned in the introduction, that make this reaction biomarker patient DNA more efficient. That means it is faster, and laboratories need to add less quantity of microliters of biomarkers to obtain the same results. So basically, with our reagents, we are not changing laboratory routine, that it is something bad for people that are working in the laboratories to see their laboratory routine changed overnight. At least for me it was, but for a lot of people that I know, it was terrible if I was jumping in the laboratory and say, okay, forget about everything you are doing right now, do something totally new. No, it doesn't work as an innovation. Just basically, we allow laboratory to continue to do the same, but adding our reagents during the DNA analysis, and they obtain this kind of nice reduction of time, of cost, while keeping the sensitivity of the test. Additionally, in the automation, we are still working also to bring our innovation in the automation. As mentioned, first was the introduction of silver in situ hybridization, but it is a process that is long, so more than 1 working day. So the need to improve the automatic process for us is to make possible to carry on fluorescent in situ hybridization on the device, but also in less time. And we accomplished that as a in part. So we started just 1 year ago to do that. Right now, we allow to perform an in situ hybridization in a semi-automation way in just 4 hours. So as mentioned, and without increasing costs. So basically, our innovation, again, is not working to disrupt totally or to add something new in a process that is already solid, let's say, in the routine. And it works for every kind of innovation you would like to bring into the diagnostics procedure. So there are procedures that are approved, that are suggested in the international guidelines. So it is easier to innovate and implement by implementing something that is already approved and consolidated in a patient's algorithm of evaluation than to say I have innovated a brand new way to diagnose genetic alteration that will replace everything. This is right now in the diagnostics pipeline. So it is a great innovation. It will be a great innovation. However, people need to be aware that it requires not only a long certification process, but the acceptance from the international medical community that, for my experience, at least in Europe, is not so innovative or not so prone for disruption innovations. But that if you add small bricks into the innovation building, let's say it is easier to penetrate that market. Next step for in situ hybridization, which is not so next in the future, but it is something that it is already on the market, but not for diagnostic usage, is the in situ sequencing. In situ sequencing is basically, as you can see in that image, so it is performed on the big samples, like macro section. So where you take all the let's say surgical sample, which can be 5 centimeter for 5 centimeter or more, you cut in slice, and then you do hybridization on that. The small bright dots are where the gene is present. Let's say the marker is present. This is what we did in collaboration with another company that now has been acquired, but it was a Swedish company. And basically, the in situ sequencing is a technology. So forget about I can eventually share the slide, so don't need to take note on that. But just to make it that slide more simple, in situ sequencing is a techniques that was developed at the beginning during the 80s at I guess Yale University, if I'm not wrong, but in the US, that at that time, they are trying to match the in situ PCR. So basically adding on the tissue slide, not in a vial like that, on a tissue slide, they are adding primers and enzymes to carry on a PCR, and then they are matching on the PCR products, always on the slide, a fluorescent, probe like for the in situ hybridization. Right now, the in situ sequencing has been evolved. Right now, they are performing with the ring, let's say circle DNA products. So it is a PCR that works in circle like for a lamp PCR, and they are intercalating fluorescent dye to detect different signals. So you can detect a lot of genetic markers in the same section, as you can see in those picture, but with the localization also of the genetic expression. So you can see that there is a blue gene in the upper area, the violet in the middle, the yellow in the bottom right and bottom left, and so on. And it is very important because with the standard PCR, you cannot do that. Or with NGS, you can try to do that, but it is difficult to localize cell by cell the product of amplification. And yeah, basically, this is the last, the new frontier, let's say, that will be in the in situ hybridization. To me, in situ hybridization will remain the gold standard technique for in cancer diagnosis, and especially with the implementation of target therapies. There will be more need for in situ hybridization, because despite NGS can identify a huge number of very single base mutation, there is still the need to localize precisely cell by cell where the mutation is, and which is the mutation, the relationship between mutated and not mutated cell. So having a single cell resolution in situ, so on the slide, it is a pro that still keeping in situ hybridization alive. Just an anecdote, I started doing in situ hybridization in 2003, so I'm that old, and year by year there are some sales specialists from a PCR company or NGS company came to my lab and say, "Oh, from next year the in situ hybridization will be replaced by Sanger PCR, or will be replaced by real-time PCR, or by NGS, or by that, or by that." By the way, day by day, year by year, new guidelines from the World Health Organization for cancer detection and diagnosis and patient management includes new marker with in situ hybridization. So it's something that will last longer, I hope also. But in situ sequencing can be a powerful technique once it will be cost affordable and also technically affordable, because right now you need one test cost a huge amount of money and require instruments and big piece of instruments, very complicated techniques. Just a small take-home message, so I leave you space for question after that. As mentioned, in situ hybridization is still and will be a gold standard for molecular technique for cancer diagnosis. Obviously, we are addressing human healthcare, but in situ hybridization is penetrating the veterinary oncology, and also it can be used, or it was used, but with minor application to detect viral RNA during COVID pandemic, for example, to detect at the beginning where the virus migrate into the lung. So they use in situ hybridization for RNA to detect which cell was colonized by COVID. So is an example. So in microbiology, it can be used. Already, so available innovations are represented by automation and innovative reagents. And as mentioned, we can there will be space to innovate let's say, in situ sequencing, because it is something new. The working in situ sequencing is something new. So there is space for innovation. And to me, to allow a more flexibility, a higher flexibility and affordability to automation. And again, this is a trend that it is not working also only for in situ hybridization, but bigger is a company, slower is in processing everything, and lower are the risk that they are keen to take. So that means that very big company internally are not innovating enough or so fast. So recent innovations in situ hybridization, but also in pharma and in mainly any healthcare sectors, are driven by small companies, startups that are often originating from academic institutions. Industry leaders right now are crucial in supporting startup to stay alive and to bring innovation to the market, because they are the adopters of that innovation. And yeah, basically that's all for my presentation. I will be happy to reply to your question. If you have right now is fine, or please take my email or my contact from the organization and send me question by mail. Happy to reply. Thank you.
Is there any questions from participant? It's a really very insightful session, sir. You have cleared our concept of in situ hybridization, how it is useful in cancer diagnosis and all.
Thank you.
Thank you for guidance. Yeah.
Thank you very much. Thank you very much. Again, feel free to share with the attendant my presentation if they are willing to read it, and to contact me after that meeting also for question and so on. Happy to support. And thank you.
Thank you, Dr. Enrico. It was a really nice session. Um, I think I already asked you questions on WhatsApp previously. So we are actually typing with some of the hospitals. We will be having some, cancer patient issues, and we are interested to utilize, the your kit. But also we are doing animal studies. So for that purpose also actually we want to use. So my question is that can we actually utilize for both tissues, animal and for the human tissues?
Yeah, let's say so. Our kit, yes. So they are agnostics, let's say, about tissue and marker. If you do in situ hybridization, regarding probes, we may support also with biomarkers. For human, for sure. For animal, it depends on the marker, because if there is an affinity with the human sequence, we can use the same. So we have experience with HER2 gene amplification in dogs and cats by using human probes. For ALK, which is receptor protein for detected for lung cancer, we have the same affinity. or other gene associated to sarcomas. It is case by case by animal, and we can support it for kit for sure. So if you have your already have your probes and your reagents, we can be complementary. Let's say.
Um, and this the procedure which you explained was on the slide, right? Um, is there like in the market, or are you actually planning to develop something in the 96 well plate, or
So
maybe 8 well or 6 well?
yeah. So let's say if they are cells, yes. So I mentioned the slides because for in surgical pathology is the most common support that is used because you cut the paraffin block and put on the slides. But we did also on plates on wells with the cells growed on those plates with the 96 well. So any kind of cell can be tested. Obviously, the protocol will be different, but there is no. We have also a protocol for cytological samples and so on. So there is not an issue.
Okay,
Really.
so the cells are the one. With the tissue, you can actually, when you cut the tissues, then only it can be used. But the 96 well plate be very small.
Yeah,
Maybe the 6 well or 4 well plates can be utilized.
yeah, it will be better. Also, if you have there are some slides that we used in with that had have 8 small wells, but on a slide. So you grow that cell on a slide, and then once you need to test, you just take out the wells and then fix like in a cytogenetic fix them and proceed with the test. So this is the best support for us.
Okay, and so like when I was in UK while doing my PhD, I was actually also looking at comment essay with fish,
Okay.
where you can look for. I think Joe is also there. I will add Joe. She can also put her some input. So
Afternoon.
we. Hi, good afternoon, Joe. Hi.
How
So
is everybody?
yeah, I'm
Hi.
fine. Thanks. How are you?
Good, thank you.
So I was just asking Dr. Enrico the question. Actually, we tried to do comment essay with fish. So their kit is actually for the fish. So with comment essay with fish, you can do DNA damage as well as chromosomes positioning.
Yeah.
So what is your thought on that? That when you are working on the fish, are you actually also looking for DNA side as well?
I mean, we're looking at structure, sort of doing super resolution stuff now, but I can't say, Pravin, that we've done comet fish yet. I haven't. We're still just doing fish.
Fish. Okay.
Yeah.
How How about you, Enrico? What you can? Uh,
So we regarding chromosome, so we never experienced that because we didn't have at my hospital enough budget or instruments to perform the chromosome painting, let's say, with the in situ hybridization. So, and also in Italy, for instance, I don't know many centers that did chromosome painting. To be honest, it is a pretty probably cost less an intern that do standard G bending and stay hours cutting chromosome on a PC that the apparatus to do chromosome painting with in situ hybridization right now. So I believe so. But so we did fish, and so let's say structurally we can just check for rearrangements, duplication, and so on. So if for minor, it's probably Prof. Bridger know better than me. For minor alteration, let's say it is still a suitable technology. So
okay.
Yep.
Thanks,
thanks.
Enrico. Over to you, Swarda madam.
Yes, sir. Now I request Rutuja Shinde ma'am to propose vote of thanks. Yes, over to you, ma'am.
Thank you, Swarda ma'am. Am I audible?
Yes, ma'am. Yeah, you're audible.
Yes.
Yeah, okay. So, as
You're not audible, ma'am. Now, Rutuja, ma'am.
am I audible, sir?
Yes, yes. Now you're audible.
Okay, just a minute, sir. Am I audible now?
Yes, yes, ma'am. You're audible.
Again, you are not audible, Rutuja ma'am.
Hello.
Yes, now you are audible.
I think there's some problem. Now,
Yes, you are audible. Again, there is some problem. I think she's facing some network issue.
hello.
Yes, you are audible, but when you are starting to speak now, you are not audible.
Okay, so as we are wrapping up this insightful session, I want to thanks to Dr. Enrico D. Otto for your valuable time, sir, for this second annual conference of Swalife, and whatever you have gave the information on the biomedical technology that was so useful for the participants also and for the research seekers also. So thanks a lot for your valuable time. Thank you, all of you. And over to you, Swada ma'am.
Yes, thank you, Rutuja ma'am.
Thank you.
Now
Thank
we
you very
will,
much.
yeah, thank you, sir. Good day, sir. Yeah. Now we will move towards the next session, which is on alterations to genome organization in aging and premature aging by Dr. Gianna Bridger, ma'am. So I request Ms. Rashmi Yadav, ma'am, to introduce our guest speaker. Over to you, ma'am.
Do you want me to put my slides up?
Okay, ma'am.
Can you see that?
No, ma'am.
Okay, so I'm sharing.
Yeah, now it's visible.
Oh, it's come. Okay, because I can't now see you guys. So um, if you just tell me when to start, then I can go ahead.
Yes, ma'am. We will wait 5 minutes for that,
Okay.
ma'am. Just a minute.
Hello, Rashmi madam. Can you hear?
Yes, sir. Am I audible, sir?
Yeah, yeah, you're audible.
So shall I start?
Yes,
Yeah, yeah, you
yes,
can.
ma'am.
Yes. Hello, everyone. This is Rashmi Yadav. Now I'll be introducing our speaker for the session, Dr. Joanna Bridger. Ma'am holds a dignity of Director of the Center for Genome Engineering and Maintenance at Brunel University, London. To talk about ma'am, she holds an expertise and just a minute, I'm just logging. Yeah. Ma'am's experience. Ma'am is with Brunel University London since 29 years. She works as a professor of genome organization from 2020 till present date. She also worked as a director for the Center of Genome Engineering and Maintenance since 2019 till present date. Ma'am also worked as a reader associate from 1999 to 20. Ma'am has a non-clinical postdoctoral scientist in MRC Human Genetics Unit at the University of Edinburgh. She was a postdoctoral researcher at DKFZ German Cancer Research Center. With this introduction, we have ma'am's expertise in different areas and she'll be dealing with alterations to genome organization in aging and premature aging. This was a very brief introduction to tell about ma'am. We would like to enjoy your session. Over to you, ma'am. Thank you.
Thank you very much. Thank you very much for inviting me to this special day. I have known Dr. Bade for a long time. He was a PhD student at Brunel University London with myself and my husband, Dr. Ian Kill. And he was a tour de force, I tell you. I guess he still is. He was just pushing all the time, coming up with great ideas and pushing forward. So anyway, I'm here to tell you about our work that we've been
Thank
working.
you, Joe.
Oh, you're very welcome. We miss you. um, yes, so I'm going to tell you about Brunel University. So sometimes people think that Brunel is in the Middle East, or it's in a town called Bristol, a city called Bristol in the UK, because it's named after a very famous engineer from Bristol. But it's not. It's very near Heathrow Airport, on the outskirts of London. And here I've got, I hope you can see them. I'm sure you can. So I've got the map here. Can you see my cursor? And then we've got this is the lecture center. Okay. Most of Brunel well, okay. Half of Brunel is, chrome and glass, very modern buildings. The other half are these concrete buildings from the 1960s. But this building is quite special because it's what's called listed. So it's protected. Nobody can knock it down because it's a really classic um, representation of what's called brutalist architecture. And many films have been filmed in here, so we keep it, and I love it. Other people hate it. And as the introduction said, I am the director of the Center for Genome Engineering and Maintenance, which is quite a mouthful, I know, but we wanted to call it this because it can be then just CenGem. And CenGem is much more easy to say. So these are the groups that are in CenGem at Brunel University. So we have a range of different disciplines and interests and a number of different people. So we have 32 different group leaders within this center. And I think, Pravin, that's grown quite a lot since you left. So it's something that is definitely growing. So we have
Yes,
cancer.
yes. Sorry, Joe. I think you're on the second slide or the first slide.
Second slide.
Yeah, I think the slides are not moving. Then
Okay.
maybe you.
I can stop sharing and start again.
Yeah, yeah. Stop sharing.
Okay.
Yes, please. Yeah.
Okay. Share screen. Share screen. Can we see now?
Yes.
Is it a problem if
Yeah.
I go to the full if I go to full presentation?
Yeah.
Otherwise, I can see.
I, yeah. Yeah. Go for the full presentation.
Okay, can you see that now?
Mm, no, I think that when you are doing the slideshow, that is
Okay,
different screen, and the one which
okay.
you are sharing is different. Yeah.
Is this okay?
Yeah. Yeah. This is fine.
I'll keep it like this. Let's go with what's working.
Yeah.
Obviously.
Yeah.
Right. So yeah, we have cancer mechanisms, lots of different cancers, genome organization, which is where we're what we're talking about today. Disease and therapeutics. So this is gene therapy. So there's a lot of biologists at Brunel that work on gene therapy and genome editing. Organ on a chip group, which is growing very, very quickly, and I'm hoping this will become a center of it in its own right. Cellular differentiation and tissue structure, genomic informatics and epidemiology. So a lot of the bioinformatics is in there. And we've just started a big network working on how cells and tissues, etc., behave in space in microgravity. So that's a new interest of ours. So let's go to genome organization. So the genome here is shown as this is a human karyotype with all the different chromosomes, and you can see that they're all different sizes and they've got centromeres in different places. And all this DNA from these different chromosomes has to be organized and placed inside the cell nucleus. So here's the cell nucleus, like a bowl here. And it's not all higgledy-piggledy like a bowl of spaghetti, as I still see represented in certain textbooks and that. And it's much more organized than that. It's much more like here we have some ravioli, spaghetti, pasta. So you can see that it's not like spaghetti. It's actually more like these areas, these structures, we call them territories. And what we do in my laboratory, although there are lots of different ways of studying genome organization inside a cell nucleus, we paint chromosomes. So we're painting them with their absolute direct copy of their DNA that you see in each chromosome. So it hybridizes very specifically, and then you can pick out these individual chromosomes very easy. So here in this bottom picture here, we have individual chromosome territories. So that's already a level of organization that we see inside the cell nucleus. And here we have a nice cartoon. So this is this is the cartoon of all the chromosomes inside a cell nucleus. But we have to think about what else is in the nucleus that's helping this genome organization. I will get into this in a little bit more detail, but we have the nuclear envelope around the outside here with lots of different proteins that bind to each other, but also bind to the DNA, the chromatin, the chromosomes, even anchoring them at the nuclear periphery here. And there's lots of other structures within the nucleus, like the nucleolus, more towards the center, and also nuclear bodies that have chromatin anchoring or binding capabilities. This is a beautiful picture. It looks like it's a cartoon being painted, but it's actually a representation of when you paint all the different chromosomes from that karyotype in a cell nucleus, and then you pseudo color them different colors. So you can see that there's a little bit of touching and maybe overlapping, but they're all sitting in their own individual territories. And these are all the chromosomes labeled here. So we've got, 18 here, the Y is here in the middle, there's an X at the edge, 19 here in the interior. So all different colors. And you can do this by labeling up the DNA probe you're going to do the fluorescence in situ hybridization with, called FISH, with different markers or haptens that we call them, like biotin. We use biotin, we use deoxygenin, we use DNP, dinitrophenol. So and then you can do combinations of those. So you can have biotin with dig, you can have biotin with DNP, and then with all those combinations, you can end up painting all the different, or pseudo coloring all the different chromosomes. So it's quite a complicated system, right? I mean, it's not just everything just ends up in a place where it lands. It's organized. And as cells come out of mitosis, they are the different proteins are finding different parts of the genome due to their epigenetic marks and taking them, pulling them to different places in the nucleus. So there's a lot of movement and organization going on just the first few hours after mitosis. So there are a range of techniques to look at genome organization, and this is a nice figure that I've just downloaded from another paper. But it does show all the different ways of doing it. You can use FISH, fluorescence in situ hybridization, which is where we paint the individual chromosomes. And you can do that in flattened cells, or you can do it in three-dimensionally preserved cells. And that's what we do. It's now a capability to use super resolution. So this can, we've got a super resolution microscope now at Brunel, which was hard fought for. It was a long time coming, but we do have one now. And so you can then even go down another level of magnification to look at the chromosomes. But there's all sorts of other global studies where you can then take out bits of DNA that sit next to each other or are associated with different structures in the nucleus, say at the nuclear envelope, at the nuclear edge, and then do the sequencing. And then by taking that sequence data and then using computer, you can then rebuild what things are sitting next to each other inside the nucleus, where they are in the nucleus. And so that's that's a good way of doing it, as long as you have plenty of money and you treat your cells very well. And also you have then mixed populations. So you have heterogeneity within populations of cells. So all these techniques are done using samples of cells that have all sorts of cells in different phases of the cell cycle. They might also be non-proliferating versus proliferating. And so you're looking at the sort of big picture of that in a culture, rather than doing single cells like you do with fish. So I have to say, although we are trying these different techniques and we're using something called DAM-ID, where we can look at DNA or chromatin that's a binding to certain proteins in the nucleus, we do stick with the fish, and it's sort of coming more into vogue again, I have to say. And you can do more, because it's not so expensive, you can look at more time points, if you see what I mean. So every time you want to sequence a cell culture, it's like £5,000, which is quite a lot. I mean, if you're a rich lab and you've got lots of grants, then okay. But we can then do time points that are 5 or 10 minutes difference, and that allows us to look at things that change very quickly. Okay. When you do use these techniques, and people have compared looking at the global distribution of the DNA, the chromosomes, the chromatin, to how it looks in fish, and the comparison is mostly favorable. They fit together. So it's important that these two techniques aren't seeing different things. It is also important that when you have cells that you're going to do these global techniques on, that they are treated correctly. You don't put them under stress. You don't give them a cold shock, like putting them on ice before you take the DNA out, those kind of things, because that will change and move the chromosomes and genes inside the cell nucleus. So this is kind of what we do. So this is, I know it's almost like we've been doing this kind of distribution or analysis for a long time, but it works, and we know different studies, different people, different labs, and it keeps working. So we stick with it. Don't fix what ain't broken, as they say. But we are looking at other techniques as well to compare and keep things in modern. But I do have a PhD student that is actually not in my acknowledgements, who is rewriting this analysis for PC, and we're going to be able to share it out, because it's actually very difficult to get this working in other labs but we want to be able to do that. So what we do is we have cells, and this is a cell nucleus here, and there's another one here. So what you can't see is the cytoplasm here. So they've been fixed either to make them totally and utterly two-dimensional. So they're flattened completely, and then we do the fluorescence in situ hybridization on them with the probe of interest, or we fix them for to preserve their three-dimensionality, and that's we use cross-linking fixatives like paraformaldehyde and then puncture holes into the nuclear envelope with various detergents. So what we do is we've done the comparisons between these two different fixations and analyses over the years, and it comes out very similar. Most of the time it's fine. There was just one gene that looked like it was in a different position when we compared 2D to 3D. So with the 2D, it's totally flat. Here we've got the chromosome territories in green, and we've got the nucleus outlined here. That's what the computer does. And then it erodes in, creating 5 shells of equal area. And then you can see these 5 shells. And then it measures the amount of chromosome signal or gene signal from the fish, and also the amount of DNA that's in each of these shells. And because you flattened something that was three-dimensional, there will be more DNA in the center. Does that make sense? So there's more DNA in the center, sorry. And I bumped my microphone. And so then we do our normalization. So we divide the signal of the chromosome or the gene by the amount of DNA that's in each shell, and then plot the graphs like this. So this would be the nuclear periphery. And this is then a chromosome that's very much at the nuclear periphery. This is one that's then an intermediate positioning, sort of somewhere here in shells 3, peaking in shells 3, and then one that's very much into the nuclear interior. Okay, so that these are handmade graphs. They're not the real thing, but you'll see some. But because we're looking at single cells, we can also use a marker that's used a lot in cancer diagnoses because it picks out proliferating cells, and that is Ki67. And you can see it here in red at the nucleoli. And in early G1, in early cells just after they've divided, it can be quite speckly, but it's still positive. And you can see this cell here, even though it's stained for Ki67, it's totally negative. So it's a non-proliferating cell. So it's either quiescent, and so it's a cell that's come out of the cell cycle into G0, but that cell can come back in again. So it's reversible, or it's senescent. So if we take away the serum, we take away its food, then it will become quiescent. But if we leave it in high serum, so it's got plenty of nutrients, but it's then still negative, and it's still got plenty of space around it to divide, then it's a senescent cell. And so we can, with this marker, we can't tell between senescence or quiescence, but there are other ways that we can do that now. So we look at different cells. So here's some fibroblasts here. We look at fibroblasts. So proliferating fibroblasts, even in a young culture, you are going to have a number of cells that are already senescent. So it's really important to know to differentiate between Ki67 positive and negative cells. And that's what we do with our fish. So we can do these comparisons. So quiescent fibroblasts, they're reversibly non-proliferating, so they can come back into the cell cycle. So they'll be Ki67 negative. So if you use young ones, there are fewer senescent cells in there. Senescent fibroblasts are where you then can grow them for many passages, because they're primary cells, grow them for many passages, and they then enter senescence, and they are irreversible. They don't come back in again, although some people are finding drugs that can reverse senescence. So that's very exciting. So they're also Ki67 negative. So we've also looked at lots of other different cells. So after sort of mapping chromosome states, we've then gone on to do cancers and other different diseases, but also then looked at other cell types as well and different models. So the snail is going to come into this today, but not the pig work. So this is a talk about aging, and it's something we are all concerned about. We are all aging. Um, um, there are a number of drugs that are in clinical trial that are hopefully going to be used to prolong health span, maybe allow people to live a little bit longer. But it is the senescent cells that build up in our body that are the problem. So there are a number of hallmarks here of aging, genomic instability, where the genome doesn't behave as well anymore. It's not as healthy, not responding to cues the same, maybe disorganized. There may be broken bits and the repair doesn't work as well. Telomere attrition. So this is where the telomeres get shorter and shorter. And it's in the past, it's always been, okay, that is the aging clock, but it's not the only story. Telomeres becoming shorter anymore. There are a number of epigenetic alterations, so changes to the chromatin structure that happen inside the cell. There are other things that happen within the body. Okay, loss of proteostasis, deregulated nutrient sensing, which is important because you can in animal models with starvation or calorific sort of preventing, not eating a lot of calories, eating reducing the calories. Certain animals have shown that they can have a better longevity, so live longer. Okay, but cellular senescence is one of the big hallmarks of aging, and if you can stop cells becoming senescent or take them out of the picture, it seems then things different animals can live longer. This was first shown in mice. So cellular senescence is a big deal in aging. And what happens is that the primary cells will divide and divide, but only a certain number of times. And then in a culture, it doesn't happen with all cells at the same time. Gradually, more and more cells will become senescent. So we have these markers. I have a P21 is an archetypal marker, so it starts getting expressed in senescent cells. P16. There's all these different issues with the genome that you can see down the microscope. So we have senescence-associated heterochromatic foci. So they're not in all cells, but they're very bright areas of chromatin that you can see, very dense areas of chromatin. Okay, TIFs are associated with the telomeres and DNA scars, but also bits of DNA, which sets off this mechanism called SASP. And SASP is very detrimental to the tissues and other cells around it. But also, we also have, I'll just put this figure in here to talk about changes to the nuclear envelope. So the nuclear envelope would be around the nucleus here. And one of the lamins that I will talk about in a minute, show you where they are. Lamin B1 is downregulated in senescent cells. And you can see here, this is a figure from a beautiful figure from another paper with concerning neuronal cells. However, you can see that there's a downregulation of lamin B1, which is important. And I told you already that the lamins are really important in anchoring chromatin to the edge of the nucleus. So here are the nuclear lamins. So let's go back to, I just want to say about SASP. SASP is expression of all sorts of different chemokines, interleukins, and things. But if you get a senescent cell, it exudes these compounds and makes other cells around it senescent. So it's like a bystander effect. And so you get the more senescent cells you get. And so it just pushes the tissue aging. And sometimes you can see people sort of, or you say they've aged overnight, but it's this, they pass to somehow a threshold with the number of senescent cells that they have. So these are the nuclear lamins, and these are at the edge of the nucleus here. So here we've got a cell nucleus, and you've got the lamins at the edge of the nucleus. And what we have are A-type lamins and B-type lamins, and they dimerize and create this meshwork of intermediate filaments at the nuclear envelope, and they are then anchoring the chromatin and the chromosomes, the genome, the DNA to the nuclear edge. So here's a nice picture of them stained in green, and the DNA here is in blue, which is DAPI. And this electron micrograph is, you can see all the heterochromatin that's at the edge of the nuclear envelope. So here's the nuclear envelope, a nuclear pore, which is like a gate, has a gap in the heterochromatin, and you can see here that it's strongly associated in the same position, and that's because it's actually bound. So we have chromatin, specific areas of chromatin called laminar associated domains associated with the lamins at the nuclear envelope. And here's an old picture, probably doesn't look quite like this, but this is one of the first pictures showing the meshwork, the basket weave of the lamina at the inner nuclear envelope. And lamin B1, as we've already seen, is downregulated in senescence, and lamin A mutants increase. So mutations in lamin A at the edge here. So we've got lamin B and lamin A at the edge increase in cellular senescence. So the SASP does all these nasty things, but it does end up pushing senescence reinforcement, making itself senescent and other cells. It does have a role. It can stop cells going into becoming tumorigenic, or it can also, senescent cells can then stimulate tumorigenesis. So it's on the whole not a good thing, but it does help with fibrosis. So senescent cells help sort out fibrosis. Anyway, back to genome organization. These are some very complicated but very nice figures from a recent review by Tom Mastelli. So we're looking at how the DNA is then coiled up into chromatin and placed into cell nucleus. Okay, and there's all sorts of theory, and we know that we have it's called architectural elements here that are anchoring chromatin or heterochromatin to the nuclear envelope. It's also the same for the nuclear bodies that are found within the nucleus. Sorry, I banged my microphone again. This and the positioning of chromosome territory. So we do know that we've got this anchorage here and in the center, but there's other types of possibilities that probably all work together. And this is what this review is about, is how do we bring all these different models and mechanisms and theories together at the moment. So we have polymer-polymer interactions, so different proteins, etc., interacting with each other. Chromatin dynamics, so things being moved around the nucleus, and I will talk a little bit about that, probably by nuclear motor proteins. Phase separation, where different, it's quite complicated, but that's how we build these nuclear bodies, and then they separate from each other or push away, repulse other proteins or other structures, allowing this organization to get set up. Okay, and then there's probably some differences between individual cells, and that's why I think it's quite important to do single cell analysis. So back to where it all started, with looking at genome organization. This is when I was doing my postdoc in Edinburgh. I'd learned how to look at genome organization or paint chromosomes in 2 and 3D in Heidelberg in Germany. But I went then into the group of Wendy Bickmore, and they were looking at these little chromosomes. The 2 smallest chromosomes in the human karyotype are 18 and 19. So chromosome 18, 85 megabases. It's very small, but it has very few genes. So it's a very low density with respect to genes, chromosomes. So chromosome 19 is one of the ones that has the highest gene density. So it has a lot of genes, but it's also of a similar size. And so these were the 2 chromosomes that were compared to start with. And it was shown that they have a difference in structure. So remember, chromosome 18 is a very gene poor chromosome. Here it is in red. So 18 is in red, and it's quite tightly packed. Nothing much going on there. 19 has a lot of genes, and it's very active with a lot of gene expression, transcription coming off it. And you can see here in green, very open chromatin. So quite different types of chromatin. And then the location. So this is quite an old picture because it's a long, it's come out of a paper that's over 25 years old. But here we have 18 and 19, 18 in red, 19 in green, 18 in green, 19 in green. Okay, so you can see the different ways that we've painted these in different colors, just to make sure that we're looking at the same thing each time. So you can see here that 18 is in red here, in green here, at the edge of the nucleus, very much at the periphery of the nucleus, whereas 19 in the interior. Okay, so this gave the indication genome organization in proliferating cells could be a gene density distribution. And then when we went on to look at all the chromosomes in Wendy's lab, in the human karyotype, indeed, they fitted the gene density distribution. And this is proliferating cells. Okay, and it's conserved across many different species. So these are just the great apes here. But if you look at other different organisms too, when we've looked at the mice and we've put chromosomes into human chromosomes into a mouse background, they also go to the relative chromosome positionings that we see in different organisms. So just to reiterate, conserved across many different species and different cell types. But then once we started looking at the non-proliferating cells, we saw something quite different. So we saw that the chromosome positions were changed. Not every single chromosome changed, and it wasn't a non-random a random chaotic situation. So specific chromosomes had changed their location. So say chromosome 18 that we were already looking at was at the nuclear periphery. It now interior. Okay, and then also chromosome 13 is a very small chromosome and has very few genes. That was also at the nuclear periphery and then moved to the nuclear interior when we were looking at non-proliferating cells. So it doesn't matter if you're looking at cells that are quiescent, that are made by removing the serum or allowing contact inhibition, or if you've then senesce these cells by serial passage, then the cells, you end up with 13 and 18 in the nuclear interior. So, but then if you then look at cells, so chromosome 13 and 18, as I said, sorry, my daughter is just leaving for a party, so I'm a little bit distracted, but I will be back on it. So we have then, there are differences between quiescent and senescent fibroblasts. So this is quite important. If we're looking at non-proliferating cells and we're interested in non-proliferating cells, and we know that senescent cells are causing aging, then it's an important study to be able to tell the difference between them. And there are differences, but only in the two chromosomes that we have found. So we have chromosome 10 and chromosome 15. So 13 and 18, the same in quiescent and senescent cells. So if we look at chromosome 10, so we're comparing here in 2 dimensions or in 3 dimensions. So we've done the fluorescence in situ hybridization FISH for both these situations. With chromosome 10 in proliferating cells, it's found in an intermediate position, which is quite interesting because then it fits with its gene density. So it has an intermediate level of gene density. And so then it's in between. But in quiescent cells, we find it very much at the nuclear edge. You can see that here. And in senescent cells, it's in the interior. So it couldn't be in two more different situations, positions in quiescent and senescent cells. And this could potentially be a marker for senescence, because it is quite difficult to tell the difference between a senescent cell and a quiescent cell otherwise, although you do have P21 markers and stuff. So this is then done in 3D, so you can see there are different distributions of the chromosome positions. So I would like to suggest that you could use interphase chromosome positioning as another marker, a hallmark of aging, and to look at chromosome 10 and to see, and 15, what the differences are in gene expression. And we know that on chromosome 10, there are a lot of genes that are involved in proliferation. And maybe these need to be shut down in quiescent cells and in senescent cells, but shut down differently, silenced differently, and that's why they end up in different positions in the cell nucleus. And so over the years, we have been, from my postdoc then to having my own lab for the last, I guess I started in 2000, 24 years, we have been mapping and looking at these chromosomes in different situations. So we started with the proliferating chromosomes 18 and 19, showed that they were in different positions in quiescent and senescent cells. And then we were moved on to quiescent cells, looking at all the chromosomes. And then recently we published the senescence work where the positioning is changed with chromosome 10. But you can see here that I'm talking about motor activity, movement of chromosomes. So if a chromosome is in a new location, that implies movement. And if we look at the quiescent cells, moving the serum, their positioning changes very, very rapidly. So the positioning, it isn't something that happens very slowly over time, or that the cells have to go through mitosis. What happens is so here we have chromosome 10 in its intermediate location. Remember, that's the periphery of the nucleus. That's the interior there on this graph. So within 15 minutes or even 10 minutes, it's moved from an intermediate position to its peripheral position. Okay, same for chromosome 13, same for 18, but X always stays at the edge. It's a great control chromosome. And it's not just a release of chromosomes from the nuclear edge, because we've got chromosomes moving different ways. So you've got chromosome 10 going that way, and chromosome 18 and 13 going towards the interior. So chromosome 10 going towards the periphery. So it implies that it is a controlled mechanism with nuclear motor activity. I need to just change my slide position. There we go. So it implies that it is look at motor proteins in the cell nucleus. I'd already heard that there could be motor proteins in the cell nucleus, like nuclear myosins, nuclear actin. There is a difference in distribution in proliferating versus quiescent versus senescent cells. So the idea is, and the other studies have looked at this, of moving genes, Andrew Belmont, people like that, have looked at the possibility that nuclear myosin 1 with its head could walk down these short actin filaments that are inside the nucleus, moving chromatin around. And this could explain why these things happen quite rapidly. Okay, so Chi67 here is showing that these are non-proliferating cells, so completely different distribution than from this one in proliferating cells, where we've got something at the edge, some at the nucleolus, but also all through the nuclear plasm. And what's quite interesting is, so we can take away the serum, we can give these cells a heat shock, and they will move their chromosomes very rapidly. However, if you're looking at a senescent cell, and you can tell the difference between a senescent cell and proliferating cell because you've got the Chi67 there, you cannot induce the change in position. So chromosome 10 here, it's an old cell. You take away their serum, it should go to the edge, but it doesn't. It's in the interior. Okay. And when we look at chromosome 11, which has the heat shock, some of the heat shock genes on it, when you give the proliferating cells that are young, we've got Ki67 positive, there is a movement of the chromosome, but in senescent cells, absolutely nothing happens. And so we believe that the motors aren't built in senescent cells. And this response could be really important in cells. So, a stress response, move a chromosome or genes to a new location where they can be upregulated or protected. And so you think that this inability to move chromosomes in senescent cells could be something that locks cells into senescence. And if we could overcome that by reactivating the movement or the motors, then perhaps we can prevent cells entering senescence or even reverse them once they're in senescence, or have senescent cells that can respond to stress. So now I come to this other organism that we work with in the lab. So we work with human cells, but we also work with this snail called Biomafilaria glabrata, which is now the molluscan model organism thanks to it's having its genome sequenced. So my collaborator in Washington, Dr. Matty Knight, Professor Matty Knight, should I say, sorry about that. She's been working with this snail a long time, and then we looked at its chromosomes. It hadn't had a karyotype, so we were able to karyotype this snail, but it took 10 years to do that, to get the chromosomes. It's such a tricky organism to work with. However, we were then able to look at its chromosome territories, and we found, yes, the chromosomes are in territories, and they also have their own particular positions to go to. Now, it's an important snail because it's the secondary host for a very nasty parasitic disease called schistosomiasis or bilharzia, where there are parasites in the water that burrow through the skin into an individual, through their blood vessels into the liver, where the hermaphrodite worms mate, producing lots of eggs, and it's the eggs that make people very sick. So the immune reaction to the parasite's eggs, and if it's untreated, it ends up in stomach cancer. However, there are treatments, but people want to work on the snail, that sort of the link between the snail and the parasite, to eradicate and block it entering humans. But what we found when we were looking at these snails, this was a paper published a while ago, but you have susceptible snails that are susceptible to the disease and resistant strain that aren't that don't get infected by the parasite. And of course, that's a great comparison to do between the ones that work and the ones that don't. So when we infected or exposed these snails to the parasite, so here I have the Miracidia, genes moved. Okay, so we've got genes moving and we've got gene expression happening. And we showed this. And actually, this was the first time somebody saw a gene move before it was transcribed, because the field was like, what happens? Which comes first? So then when the parasite is attenuated, it's irradiated. It doesn't happen. The gene movement doesn't happen. And it doesn't happen in the resistant snails. So, and it happens very quickly. So it happens within 15 minutes. So again, we thought this is one of these active movements that must be down to nuclear motor protein. And if you use, I'm not going to give it its big name, but if you use this inhibitor of nuclear myosin 1, BDM, we then stop that gene movement after a heat shock or infection. So it can be a heat shock or infection. But here it's a 1 hour heat shock, and you can see that the gene has moved. With the BDM, it hasn't moved at all. I'm going to, excuse me. I beg your pardon. Okay, so then we get gene expression from the gene that moves and is much down regulated when the gene excuse me.
All right, Joe.
Okay, I'll carry on now. I had to get some
Yeah,
water.
yeah.
Apologies. I'm getting old. Okay, so we have the snail as a great model for aging studies. Where are we? No, I haven't got there yet. Can you hear me? I have unmuted, haven't I?
Yeah, yeah, we can hear
Yeah.
you.
So
Hello.
this is then. Can you hear me?
Yeah, yeah, yeah.
Yeah. Okay. Good. 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 We're back on. We're back on target. So you can see here we've got BDM in little speckles inside the nuclei. Sorry, nuclear myosin in little speckles, and then with BDN treatment, it's not inside the nucleus anymore. So we think we have messed about with the nuclear motors and stop that gene movement. So we know we can use heat shock, but look in old cells. So these are young cells. Oh, sorry, not cells. The snails. So this is a whole organism. So you can heat shock these snails. They don't die. They just get a little bit hot. So they go from 27 to 32 degrees. But the genes move. So this is the heat shock genes moving from the interior of the nuclei inside the snails to the periphery. And then if we use old snails, beg your pardon, 12 months old, this is considered quite old as a snail because they only live to about 18 months. You can see that we don't get any G movement, that it's almost identical. And this would be a number of snails together. So in the old snails, we don't have this repositioning anymore. And although the nuclear myosin hasn't gone completely, you can see this is the juvenile snail, this is the old snail. It is reduced. And so if we could control this gene movement, we could maybe control the disease, but also it makes it the snail a really nice model to use for aging studies because it's a water snail and it's very easily druggable. So you can run a number of drugs through it very easily. And you don't need in the UK an animal license to work with it because it's a mollusk. And so hopefully we're going to be able to look to see if we can reinstate the chromosome relocation mechanism in old snails, and then come up with some compounds that are going to be really important in anti-aging. So this brings me to premature aging syndrome that we're looking at, and that does fit in with the cellular senescence and the non-proliferation of the human cells. So we're back to humans now. Unfortunately, the snail has a B-type lamin and not an A-type lamin because the premature aging syndrome here, Hutchinson-Gilford progeria syndrome, is caused by a lamin A mutation. Otherwise, we could have had a progeria snail and looked at drugs through that. But it's a lamin B, which we know is associated with aging. So that's interesting. So Hutchinson-Gilford progeria syndrome, as you can see from the images I've showed you, is a very rapid aging syndrome. So it's caused by a single mutation in the lamin A gene. And remember that the lamin A is at the periphery of the cell nucleus. And it's just one base pair change here in exon 11. But that creates a truncated protein, but also a protein that's not, that cannot be modified. So normally, the pre-lamin is carboxymethylated, it's farnesylated, and that's what causes the problem, the farnesylation. But this is then chopped off. So pre-lamin is quite toxic. This is chopped off before it gets to the edge of the nucleus, into the nuclear periphery. In progeria cells or patients, You have a truncated protein, but it also this chopping off of the farnesyl group and the carboxy end can't happen. And so you end up again with a toxic protein, and that's called progerin. So it's the farnesyl group here that makes it so toxic. And not all of the cells look like this, but this mutation ends up making the cells misshapen and have these invaginations, etc., etc., herniations. And so this obviously affects genome organization. And this is what we have shown. So the chromosome territories, the positions, they're in the wrong place, but they're not like senescent cells because we looked at chromosome 10. They look like a quiescent with chromosome 10 at the periphery, not in the interior. And so we've shown that here. But some of the drugs that the children have been on, one of the most successful ones that's been through clinical trial is a farnesyl transferase inhibitor, and that blocks the farnesylation of the lamin A. And so you still have a truncated lamin A that cannot be cut, but it doesn't have a farnesyl group on it. And so this makes it much less toxic. And so that treatment, without the farnesyl group, the lamin A seems to be able to behave better and put chromosomes back to the right locations. And we showed it twice. We went through a whole load of other drugs, but still farnesyl transferase inhibitors plus rapamycin were very good at putting chromosomes back into the right locations. But not only that, the farnesyl transferase inhibitors were able to rescue the chromosome movement. So in the progeria cells, just like the senescent cells, they couldn't move a chromosome. So you could stress the cells, but there was no movement of chromosomes. So here you have chromosome 10. Okay, and nothing is really these are the okay. So there's some difference, but when you treat with FTI, chromosome 10 is able to move into the interior when you put it into low serum. So you can reestablish the movement of the chromosomes again in these progeria cells. This is what it should look like. This is what it looks like in progeria cells. And then again with the FTI treatment and the GGTI, which is geranyl transferase inhibitor. So you sometimes find, or most times find, that if you block a farnesylation, then the cells will do a geranyl association with the protein, but you can block that too. So you can see it's a much more normal distribution, and then we get drugs moving. And so we tested a whole pile of different drugs that were going to be used on the children, but what we find is that FTIs are the best. So when we think about aging, it is caused by the buildup and the properties of senescent cells. So what can we do? We can prevent cells from becoming senescent, so using senotherapeutics, new drugs, kill the senescent cells, which you can do with senolytics, or dampen down the SASP, the senescence-associated secretory phenotype with the xenomorphics, or try to reverse senescence. So we're going to see if we can stop the cells chromosomes in the wrong location, but also want to re-establish that chromosome movement. So this is my acknowledgement slide. All sorts of different people have worked with us over the years. Pravin very much had his own project and had his own direction, so was working for his own goals at this time and has been very, very successful. But these are a number of different students and postdocs that have been in the lab and people that have helped with the work. So now I'll come back to the main screen. Stop sharing. Okay, sorry about coughing fit in the middle.
Thank you so much, ma'am. In spite of getting some hurdles, we appreciate that you successfully completed and delivered us the important information which we are heading for.
Thank you.
Thank you, ma'am, for helping us to understand the different aspects of structure, the genome structure, the importance of cellular senescence, the concepts behind the chromosomes. Also, letting us know the role of nuclear protein aging, availing us how the nucleus based aging clock works, and introducing us the new preclinical model for molluscans to identify the chromosomal study. This was something which has really got some input in our knowledge, and this was very much beautifully presented and well explained to us. Shall
Thank you
we
very
move to
much.
answer sessions? Can we have the question answers?
Yeah, yeah, sorry.
Ma'am, one question from the audience. Are there any few molecules perform satisfactory against aging in the snail model?
Have I looked at any yet? Or yeah, no, we're working towards that. Um, I have the old snails, and I mean, we've got a whole lot of different uh, drugs we want to try, but what we've been trying to do is to block the infection. So we've been using different things to see interfering with the epigenetics and things like that. But no, it's definitely on the list.
Okay. Thank you so much, ma'am. The next question is what impact does the traditional systems like yoga, Ayurvedic treatments, diet, will they be able to alter the genomics of the cell that could affect or it could participate in certain kind of treatment for premature aging or aging?
I think so with the progeria children. I mean, they definitely try different things, and they're on different drugs as well as uh, the normal, normal that affect normal aging. So like statins, lowering blood pressure, things like that. Um, they're on those kind of drugs, and they do try to limit maybe the amount of mechanical stress sometimes that's on the tissues because the cell nuclei are fragile. But um, the most success that there's been has been the FTI drugs. But now they are working towards, and it seems that it might happen quite soon, is genome engineering. So some CRISPR Cas to change the mutation, and that's where they're going for a cure. So at the moment, it's just a treatment. With normal aging, I mean, though these drugs are coming, diet very important, and restricting your calorie intake. And I think anything you can do to lower stress is very helpful because stress is very damaging. So yoga and anything like that, definitely exercise. But I think maybe if people that overdo it, then they're putting a lot more stress on their cells. So it's I guess they say adage, everything in moderation.
So this
But
we could conclude that this can be something which is a complementary or alternative treatment which
yes,
could help
absolutely.
in building the quality of life.
Yeah, absolutely. Absolutely.
Thank you so much, ma'am, for the wonderful session and availing us to interact with you. Over
Okay,
to you.
thank you very much. And I'm sorry that I seem to have a cold that I didn't realize I had.
It's absolutely fine, ma'am. Over to you, sir.
Thank you. Thank you, Professor Joe. It was actually a nice overview. I think I went back nearly 8 to 9 years when I was in the lab. I tried working with progeria cells. Uh, just you gave an overview from complete overview from fish genome organization till progeria. And when I was actually finishing my work at that time, snails were there. You were trying something with the snails. I didn't get the opportunity, but yes, it is actually a nice model, I think, for the aging work. Similar to your Sengen, we actually started Center for Drug Discovery and Development, which is CEN-DDD. It is mostly for the preclinical and clinical work. So I actually moved now a little bit further from the cells. Now actually I'm working on animals, and also some of the clinical studies are going on. Um, One of the thing which relevant to the aging, I have one question is that, there is a articles available which are talking about 3 days fasting is actually helping for the stem cells. So, like, with the aging, have you actually given any thought to do with that type of things where you can actually, uh, you know, um, stem cells can be like utilized?
I think if you can stop, sort of break with nutrition from time to time, seems very beneficial. Um, I know Chris. I don't know if you remember Chris Askew when he was
Chris,
in.
yeah,
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 so
yeah. And he's in Canada, I think now.
yeah, he is, and so he was looking at that sort of, um, looking at changing the nutrient sensing by the cells and stuff, and I think he showed that that can change the senescence of cells, so uh, prolong prolong the lifespan before they go into senescence. So yeah, I think there's something there. I mean, I've sort of haven't really worked with that, but it's yeah. I mean,
And
maybe
it's nice to,
we should do it with the
yeah.
snails. We should put the snails through fasting regime and see if
Yeah, I
they.
think that now you got the model where you can, you don't need government permission, so
Yeah.
you can give some try. We are also working on earthworm, so we are doing some work
Oh,
on
okay.
earthworm. Yeah, and
Excellent.
it's
Oh, that's interesting.
mice are there, rats are there, but earthworms are, you don't need the permission. So antimitotic activity, we actually looking into them. Also, it's nice to see Dr. Inis. She is back now, I think, right?
Yeah, she is.
Yeah.
Yeah.
So I was there when she was doing postdoc with
Yeah.
Dr. Paula, and
Yep.
now I
No,
think she is
she's now
back
a
with
group leader. Yeah. So
group
we have a
leader. Okay.
shared PhD student at the moment. So Rita. But yes,
Mhm.
Pravin, you're right. You were working with progeria cells as well. And
Yeah,
yeah.
yeah, yeah. For sure, sir.
Yeah.
We actually have started one value-added course, which is in preclinical studies, and we are utilizing bioimaging. So maybe I think we can, collaborate on that work, because we want the expertise where, image analysis can be utilized. So I will approach to
Okay.
you and come back to you on that.
Yeah, no,
So,
our imaging is going quite well. So that's just a good timing because we've just hired somebody called Alessandro Esposito, and he's world class bioimager. So the
mhm.
imaging is taking off for sure. So
So
yes,
yeah,
happy
yeah.
to, Pravin, happy to.
Nice, nice. Thanks, Joe.
Okay.
It was nice, having a
Yeah.
uh, talk with you, and also, understanding what things are going on in Brunel at the moment.
Yeah. I
Thanks.
mean it. Yeah. Okay. Thank you. Lovely to see you all. Thank you very
Nice,
much.
nice.
Take care.
Just 1 second, just one.
Thank you, Jo ma'am, for such a wonderful talk. Now I request Rutuja ma'am to give vote of thanks.
Yes,
Over to
thank you.
you,
I'm
ma'am.
audible, Swada ma'am.
Yes, yes, you are audible.
Yeah, thank you so much, Joanna ma'am. It was really a very interactive session. As we wrap up this engaging session, it is my distinct pleasure to express our heartfelt gratitude to Dr. Joanna Bridger ma'am for her exceptional contribution to the second annual conference. Your ability to explain this complex scientific information in a way that is easy to understand and impressive and has greatly increased our understanding of this important topic. We deeply appreciate your willingness to share your research work with us today. Your work is pioneering and opens new avenues for therapeutic approaches, which is inspiring us all to continue our pursuit of knowledge and innovation in healthcare. On behalf of all my colleagues and participants present over here, I extend our deepest thanks to you. Thank you, ma'am. Thank you so much.
Oh, thank you. That was lovely. That's made my day. Thank you very much.
Thank you, ma'am. Over to you, Swada ma'am.
Yes, thank you, Rutujama. So, with the permission of Bade sir, can we conclude the conference?
Yeah, yeah, we can conclude. Thank you
Thank
all.
you.
Yes.
Thanks.
Thank
Thank
you.
you, all the participants and delegates. Thank you so much. Yeah, bye.
Bye.